to discuss RNA isolation. But before we go there, let's review what is a ribonucleic acid or an RNA is. Okay, so for today's learning outcomes at the end of this chapter, we would like you to know the difference between RNA and DNA, compare and contrast the different types of RNA, recognize the importance of transcription. All of these things were thoroughly discussed in the first shifting. Today, our focus will be describing the different procedures involved in isolating RNA from different samples or specimens, and of course, appreciate the importance of RNA in the diagnosis of disease. Okay, so the outline for today, we're going to have a brief overview of what is RNA, its role in transcription, the isolation and purification of RNA, and its scientific or diagnostic significance in the clinical setting. Okay, so RNA between DNA and proteins in function and structure. It's a single-stranded nucleotide chain with local regions of intramolecular base pairing, composed of ribose sugar, and it's what you call uracil instead of a thymine in DNA. There are many classes of RNA and they have different functions. And this is the summary of the principal types of RNAs produced in the cell and their functions. So now let's go to RNA isolation, exercise 10 of your manual. So again, the learning outcomes for today will be isolation of RNA, identify troubleshooting technique, and compare available RNA isolation techniques based on the advantages and disadvantages. But before we go there, let's go do a bit of history. So RNA isolation started in the 1960s. Karl and his colleagues was able to do RNA isolation procedure using cesium chloride centrifugation technique. No, this is a method for separating nucleic acid based on the difference in density or the density gradient. No, so they are run in a centrifuge at very high speeds at 50,000 RPM for many hours. This technique is already obsolete, or there is only a few people or a few scientists using this technique. No? We have newer techniques of RNA isolation. In 1968, Kirby introduced the use of phenol and chloroform. In 1979, we have Cherdwin. He used guanidinium cyanate and beta-mercaptan ethanol, followed by ethanol extraction or ultracentrifugation in cesium chloride. And then in 1987, we have Chomzinski and Sachi. So he modified this procedure and now uses guanidinium thiocyanate, phenol, and chloroform. Talk about handling and storage of RNA specimens. In most instances, the most critical part in RNA isolation is proper handling and storage of specimens. In any way, once the sample is already degraded, there's no effective or optimized RNA isolation protocol that will be able to isolate high quality and quantity of RNA from these samples. So therefore, handling and storage plays a very, very critical role. So the following should be considered. So first, all biological materials should be handled following the universal precaution, meaning all biological materials should be considered infectious unless otherwise proven. Okay, so proper PPEs and other safety controls should be observed by the laboratory technologist. Specimens should be processed immediately. Storage, when we talk about storage, it's best stored at liquid nitrogen or at negative 80 degrees Celsius. There is what you call RNA later. No? And RNA later is an aqueous, non-toxic tissue storage reagent that rapidly permeates tissue to stabilize and protect cellular RNA in situ in unfrozen specimens. Tissue pieces are harvested and immediately submerged in RNA later for storage without jeopardizing the quality or quantity of RNA. So another important storage technique is your RNA tubes with stabilizer, or what you call PAX-gene or Tempus tubes. PAX-gene, the blood RNA system, is a marketed product for the collection, storage, and transportation of blood and stabilization of intracellular RNA in a closed tube with subsequent isolation and purification of intracellular RNA from whole blood for rt-pcr used in the molecular diagnostic setting. So there are other precautions that you should follow as well. Number one, RNA working area should be different from DNA working area if possible. Number two, gloves should be worn always and changed frequently to avoid the introduction of what you call finger RNases. No? The finger RNases were discovered in 1961 when during studies of the separation of different amino acid specific RNA, unexplained losses of activity have been observed by scientists Hawley, Apgar, and Merrill. And as a working hypothesis to explain the initial losses, it was assumed that traces of nucleases were being encountered during the experimental procedures. And true enough, after doing the experiment, after trying to identify these, we were able to identify finger RNases. So you should wear gloves, and of course you should wash your hands regularly. So number three, all tubes and bags should be capped and covered when not in use to avoid contamination with dust and other particles. Sterilized area and other materials such as your pipeters before and after working. Use UV light or spray with Lysol and alcohol. Just a reminder, with UV light, when it's on or when it's exposed, you shouldn't be in the same room, no? So that's a no, especially the ones that are selling commercially. So aside from the fact that your eyes may be damaged, ultraviolet light produces what we call free radicals that may damage cellular integrity and may actually be cancerous in the long run. So there are different RNA isolation methods. We have the organic and inorganic extraction, adsorption method or solid phase extraction methods, and use of isopycnic gradient centrifugation. And like DNA, there are major steps in extracting RNA. And the first one is what we call cell lysis. And number two, we have to dehydrate and precipitate. Number three, separation of cellular proteins and components, and precipitation and dissolution of nucleic acids. So in isolating RNA, strict precaution. This is very, very important when it comes to RNA. The presence of RNases that degrade RNA in the sample. We should be wary of many sources of that, like in your finger RNA. And sometimes in the specimen, we need to remove these so that it does not affect the RNA. So RNases can renature. So these must be eliminated or inactivated, and they remain active at a wide range of temperature, like even below -20 degrees Celsius, and can renature even after heating. So therefore, they need to be eliminated and inactivated. So one of the strategies is you need to allocate a separate RNase-free or RNF area of the laboratory. The DNA and the RNA is free. There should be two. There's a division of that. So there should be a storage of materials. Glove must always be worn in there. Disposables that come in contact with the RNA should be kept at this location and never be touched by ungloved hands. DNases or RNF articles may be used directly from their packages, and reusable glassware seldom used for RNA work. No? So after cleaning, glassware must be baked for 4 to 6 hours at 400 degrees Celsius to inactivate these RNases. Okay? So in isolating RNA, the most abundant that we get are usually your rRNA or ribosomal RNA. It's 80 to 90%, and it's the most abundant of any type of the RNAs that you know. It can be visualized by agarose-gel electrophoresis. The second one is the messenger RNA or mRNA. It's around 2.5 to 5%. It may be detected as a faint background underlying the rRNA detected by the agarose-gel electrophoresis. So please note that ribosomal RNA has small and large components, and can be, again, both of those can be visualized by agarose-gel electrophoresis. And the third one is transfer RNA and snRNA or small nuclear RNA, and these are sometimes present in the total RNA sample. So in lysis of cell or the sample or the source, there are multiple methods we can use, and you're familiar with these, right? Some of them, like your grinding, glass beading, or even simple repetitive, falls under mechanical lysis. No? So here are other considerations. If the source is a plant or an animal tissue, samples are ground to a mortar and pestle with liquid nitrogen, the first one that's in there. On the other hand, glass-based grinding may be used in samples such as eggs like nematode eggs, metacercariae of some parasites, and oocysts. These are also cases that a simple repetitive pipetting may be successful to lyse the cell and release the nucleic acid. Repetitive pipetting may be used when lysing animal cells and zooids of apicomplexon parasites, such as your sporozoites, merozoites, tachyzoites, and bryozoites, and also trypanosomal form of your Trypanoma species and Leishmania species. Okay, so those are the three lysis of sample techniques and their peculiarities. So, cell lysis for RNA isolation, we use the following materials for the organic technique. No? Number one is your detergent or phenol used in the presence of high salt, around 0.2 to 0.5 molar, or in the presence of RNase inhibitors. So, guanidine thiocyanate or guanidium isothiocyanate inactivates endogenous ribonucleases, and it also has other function during cell lysis, which is protein degradation. So it's a very strong denaturant of your RNases and can be used instead of the high salt buffers. So sometimes they add what we call 2-mercaptoethanol in the mix because it acts as a strong reducing agent. So now let's talk about protein extraction. So in protein extraction, it's extracted with phenol. So acid phenol chloroform and isoamyl alcohol in ratios of 25 to 24:1 will be able to sufficiently and efficiently extract RNA. The chloroform content denatures the protein and promotes phase separation. And in isoamyl alcohol, it prevents the foaming. No? So the organic phase should always be acidic. Therefore, if it's not in the pH of 4 to 5, it should be adjusted by overlaying buffer. So sometimes you also add RNase-free DNases directly to the isolated RNA at the end of the procedure to remove the DNA in the isolated RNA. A precipitation. So the supernatant will contain the RNA, and RNA is precipitated by addition of 2 volumes of ethanol or 1 volume of isopropanol. Glycogen or yeast transfer RNA may be added to the mix to aid in what you call a white pellet formation, which will appear after centrifugation. And this is the total RNA. No? So, the RNA precipitate is then washed in 70% alcohol to remove the coloring produced by the residual salts. So, and then resuspended in RNF buffer or water. So, in the phenol chloroform extraction, we expect that the upper phase will consist of cell lysate and the nucleic acids, which will then be transferred to another tube and mixed with isopropanol or ethanol. The white ring, which is the precipitated protein, and the lower phase, which is the chloroform. So we have what you call guanidinium thiocyanate phenol chloroform extraction, and there are many preparations available like your trizol trifast or TRI reagent. This type of commercially available reagent are able to disrupt the cell, denature the protein, and deactivated unnecessary nucleases. And the principle behind this is that RNA is separated from DNA after extraction with acidic solution containing the guanidium thiocyanate and the phenol. So you expect this kind of formation. On top is the aqueous layer of the RNA, in between is the interface consisting of the DNA, and the bottom the organic phase containing the proteins and the lipids. So chloroform is added after centrifugation. Please take note, okay? That's after centrifugation to produce this kind of setup. So the use of hot acid phenol is used for RNA from microorganisms, and cell lysates mixed with acid phenol are incubated at 65 degrees Celsius for an hour. So let's watch this together.
The Trizol Plus RNA Purification Kit is based on Trizol reagent lysis technology and PureLink silica membrane purification technology. This combination provides highly pure RNA even from difficult samples like fibrous tissue. For your protection, always wear a lab coat, gloves, and safety glasses. Prepare your work area and wash buffer, and gather your materials prior to starting the procedure. To get started, prepare your lysate. You can start with 50 to 100 micrograms of fresh or frozen tissue or cells. Add 1 ml of Trizol reagent. Homogenize thoroughly using a tissue or rotor stator homogenizer. Let the sample stand at room temperature for 5 minutes. Next, add 0.2 ml of chloroform and shake the tube vigorously for about 15 seconds. Then let the tube sit for 2 to 3 minutes at room temperature. Centrifuge your sample at 12,000 G for 15 minutes at 4 degrees Celsius. You will see 3 different layers in the tube. The colorless upper layer contains the RNA. Transfer 0.4 microliters of the upper layer to a fresh RNA-free tube and add an equal volume of 70% ethanol. Mix well by vortexing. Next, run your isolated RNA solution through a PureLink silica membrane column provided in the kit to wash away contaminants. Transfer 700 microliters of the sample to a column, which is then inserted into a 2 ml collection tube. Centrifuge at 12,000 G for 15 seconds. Discard the flow-through. Wash your bound RNA by adding 700 microliters of wash buffer 1 and centrifuging at 12,000 G for 15 seconds. Discard the flow-through and the collection tube. With the silica membrane column in a fresh collection tube, Add 500 microliters of prepared wash buffer 2 and centrifuge at 12,000 G for 15 seconds. Discard the flow-through and repeat. Dry the silica membrane by centrifuging the column at 12,000 G for 1 minute. Discard the collection tube. To elute your RNA from the silica membrane, insert the column into the recovery tube. Then add the appropriate volume of elution buffer based on your starting amount. Incubate at room temperature for 1 minute. Next, centrifuge the column inserted into a recovery tube at 12,000 G for 2 minutes. Your ultra-pure RNA is in the recovery tube, ready to be used or stored at -20 degrees Celsius.
The method of extraction of total RNA is an adsorption technique, no? So in this, we utilize the ability of RNA to form linkages to specific surfaces like beads or tubes like magnet, polystyrene, latex, cellulose, matrices, or glass fibers. And to date, this technique is the most commonly employed in commercially available RNA isolation kits. Okay, so we'll now watch another video showing this technique.
In this video, you will learn how to achieve rapid quantitative recovery of total RNA in both manual and automated workflows with the MagMax Mirvana Total RNA Isolation Kit. Throughout, please refer to the product manual for specific protocol details. Bead-based isolation is very similar to filter-based isolation. Both follow the same principles of solid phase extraction, but bead-based technologies, like that used in the MagMax Mirvana kit, allow for high throughput isolation. For manual RNA extraction, in this case from mouse liver stored in RNA later solution, first weigh and cut tissue into usable 30 mg pieces and place in a fresh aliquot of RNA later solution. Next, prepare the lysis buffer mix. Combine the lysis buffer with 0.7% tumor captoethanol in 5 ml culture tube. Lightly blot your tissue pieces to remove excess RNA later. Add your tissue sample to the lysis buffer mix and homogenize for 30 seconds. Then place your sample on ice and let the foam subside. Now prep your reagents. Preheat elution buffer to 37 degrees Celsius. Add alcohol to each wash concentrate. Combine beads and lysis binding enhancer and place on ice. Combine denase with denase reaction buffer and place on ice. Plasma samples. First, prepare your wash solutions and denase mix as described for manual isolation. After thawing your samples, add 100 microliters per well to the MagMax Express 96 deep well plate. Next, prepare your PK digestion mix. Add it to the samples, cover the plate, and shake. Incubate the plate at 65 degrees Celsius for 30 minutes. Now add 100 microliters of lysis binding mix and 20 microliters of RNA binding beads and shake for 5 minutes. Add isopropanol and shake for 10 seconds. Load your prepared plates into the Kingfisher Flex instrument. When prompted, add the rebinding reagents to the denase plate. Put the plate back on the Kingfisher Flex instrument and resume run. At the end of the run, your high-quality RNA sample is ready to be used in rt-pcr or in another downstream application. Or it can be stored at -20 degrees Celsius for later analysis.
Polyadenosine messenger RNA, no? So in this, the isolation of polyamino messenger RNA, it usually involves mostly eukaryotic mRNAs. Because remember, RNA molecules extracted from bacteria or prokaryotes lack polyadenosine tails. The method so far that's used for those are usually involving removal of non-mRNAs rather than selecting or binding the mRNAs directly. So, to enrich the yield of mRNA, especially rare transcripts, protocols employing oligomers of thymine and uracil immobilized on a matrix resin, either in a column or beads, are often used. So, this is an example of the bead or column with multiple oligomers of thymine. Sometimes it's uracil, and this is the matrix resin column, no? So, and this is the mRNA. So, the thymine tail or the polyuracil tail will bind the polyadenosine tail found exclusively on the mRNA. So, after washing away the residual RNA, the polyamino RNA is eluted by washing the column with low salt buffer containing the detergent. So, from this, around 30 to 40 nanograms of mRNA can be obtained. So, this is the graphic representation of that process, no? So, cells, RNA sources like cells, after the lysate is prepared and the cells are filtered or tissues homogenized, or sometimes total RNA. So, they bind to oligo-DT beads or sometimes magnets and incubated and spinned for 5 minutes. And then they are washed away and spin again up to 3 times, and then eluted, incubated, and spin twice. Then after this, you have your pure mRNA. Okay, so there are 2 methods commonly used in the selection of polyadenosine RNA column chromatography. One on oligo-DT columns and batch chromatography. So, column chromatography is normally used for the purification of large quantities, like greater than 25 micrograms of polyamino positive RNA isolated from mammalian cells, while batch chromatography is the preferred method when working with small amounts, less than 50 microgram of total mammalian RNA. It can be used when many RNA samples are to be processed. Batch chromatography is carried out with a fine grade of oligo-DT cellulose at optimal temperatures for binding and elution. So, there. So, this is again the bead or the column containing the polythymine tail, and this is your mRNA with the polyadenosine tail. No? So after washing away the residual RNA, polyadenosine RNA is eluted with washing the column with warm low salt buffer, as I mentioned, which has detergent, and we're able to obtain around 30 to 40 nanograms of the mRNA from 1 microgram of the total RNA. So there's another extraction of RNA, which is the isopycnic gradient method, and this method is the method of choice for isolating RNA free of proteins, DNA polysaccharides, and other cellular components. Sample is dissolved using GTC. So dissolved samples will undergo equilibrium gradient centrifugation on cesium trifluoroacetate or in cesium chloride. So that chloride or the trifluoroacetate here is used to inhibit the RNases present. No? RNA fraction then is prepared by precipitating with ethyl alcohol. In the purity, quality, and quantity of the RNA that was isolated. And there are many techniques doing just this, for RNA isolation. And please refer to exercise 11 for the instructions on the procedure. So for the learning outcomes of this, at the end of this exercise, I want you to know or determine the quality integrity of the isolated RNA. We want you to interpret the results based on accuracy and precision, analyze RNA quality and integrity, and identify potential application of RNA isolated based on the quality and the quantity, and perform QC or quality control checks for RNA. Again, there are many sources of RNA, and depending on the source, there's an expected yield, no? So, blood, buffy coat, bone marrow, the RNA yield on this is dependent on the WBC count of the patient. So, in cultured cells, the RNA yield will depend on the type of cell and the condition of cells. Liver, spleen, and heart tissues yield more RNA than brain, lung, ovary, kidney, or thymus tissues. So, isolation of RNA from fixed tissues with fixatives, is especially affected by the type of fixative use and the age and the preliminary handling of the original specimen. And again, and of course, for bacteria and fungi, depending on the type or the specific bacteria or fungi, it depends on that whether you will yield a more or lesser RNA. Again, it's very important to consider specimen handling, because as I previously discussed, specimen handling affects RNA yield, especially when we have yung RNases, no, from finger RNases. So also, as in the discussion with mRNA, usually polyadenosine mRNA will result in a much lower yield. So there are many methods of determining purity and yield. We have UV spectrometry. The nucleic acid sample is placed in a quartz cuvette, which is then placed inside the UV spectrophotometer. So the drawbacks of this technique is that it requires large sample volume, around 50 to 75 microliter, and it cannot distinguish between DNA and RNA, double and single strand. And of course, there's the risk of biological contaminants. Nanodrop spectrophotometry or spectrometry uses the same principle as your conventional spectrophotometer. It uses fiber optic technology and natural surface tension properties to capture and retain minute amounts of sample independent of traditional containment apparatus, such as the cuvettes and the capillaries. And it requires much reduced volume, like around 1 to 2 microliter compared from your UV spectrometry. Please take note of the absorption peak of your DNA and RNA at 260. Proteins are at 280, and the buffer, the trophic salts, are at 230. So the purity ratios are important for determining protein contamination or pleiotropic or buffer salts if there's too much, no, or it's contaminated with this. So purity ratios for A260 and A280, if it's less than 1.8, there's probably protein contamination. And for purity ratios of A260 and 230, they're at ratios of less than 1.4, we expect that it might have suffered from salt, meaning purity and yield. Fluorimetric method is another of those nucleic acid quantification techniques and is widely regarded as among the most sensitive methods available because DNA is naturally colorless. The principle of these methods is that the dyes used can intercalate and bind nucleic acids to non-specifically or selectively bind certain types of nucleic materials. After the dyes binding to the nucleic acid, dyes are excited at one wavelength of light and emit another wavelength of light. Therefore, different concentrations will show different intensity. Difference in spectral characteristic of nucleic acid bound fluorophores allow sample concentrations to be determined. Use dyes for fluorometric methods or your ethium bromide and Hesch 3258 and Pico-Green. So another important method is your real-time polymerase chain reaction or your quantitative polymerase chain reaction. So in this, fluorometric probes are used to bind target DNA during the annealing phase of PCR, which are eventually cleaved, and they will allow fluorescent dye emission upon target extension. So this is an example of the reaction in the real-time PCR. Please take note of these. No? So in real-time PCR reaction in which forward and reverse primers anneal to denatured DNA templates along with the reporter probe, this one, composed of a fluorophore and a quencher. No? So this fluorophore and quencher will eventually be acted upon by DNA polymerase nucleic reactivity, which cleaves the probe and freeing the fluorophore. And this fluorophore will allow fluorescent detection of DNA amplification. So there. Let's go to measurement of nucleic acid quality and quantity. So as we all know in the previous slides, nucleic acids absorb light at 260 nanometer through the adenine residues, and the absorptive constant of RNA is around 40 compared to that of the 50 in DNA. So this relationship is described, the concentration to absorbance relationship is expressed as A equals A, B, and C. The big A stands for absorbance, the small a stands for the molar absorptivity, and B for the path length, and C for the concentration. Remember that the absorbance is directly proportional to the concentration of nucleic acid in the sample. The more nucleic acid there is, the higher the absorbance. So to determine the concentration, we have to multiply the spectrophotometer reading in absorbance units by the appropriate conversion factor. So concentration significance of RNA isolation. So in RNA isolations, we're able to identify novel biomarkers for diseases through RNA isolation. mRNA expression may show the overall cellular activity at the molecular level as well. And through this gene expression studies was able to be done in diseases like your tularemia, uncomplicated malaria, dengue, breast cancers for identification of HER2 new genes, and tuberculosis. No? So right now we're doing Gene Expert in the public health centers. You know that? It's already available, no? Using Gene Expert, we were able to diagnose, because the previous methods in diagnosis tuberculosis are less specific and less sensitive, no? Like your sputum smear, no? You will learn that in microbiology. So, of course, RNA quantification, like your reverse transcriptase, quantitative polymerase chain reaction, no? So, RNA isolation is important for this procedure, the rt-pcr. So, these are important. You need to know this as well, no? The effects of tissue fixative. Sometimes you're gonna receive specimens, and will they be able to produce the nucleic acid that we want, no? So, first, among the fixatives, 10% buffered formalin, acetone, and hope, no, are good for nucleic acid quality. And B5, carnoids, Zenkers, and Bowens are less desirable types of fixatives. So these are important considerations when choosing a tissue fixative. If there's the next thing that we're going to do is watch the demonstration on non-drop spectrophotometry. We've uploaded in the Blackboard the videos related to this. So class, please watch and then finalize the related exercises. So that's about it for RNA isolation in the laboratory. Thank you.